Hi, Michelle Glass here. Welcome back to Chapter 10, Lecture 10 lecture series. Our topic here is the Neuromuscular Junction, or NMJ. Now here begins the first of our really complicated processes that we need to learn here in Chapter 10. So we have lots of steps, and you are expected to know in detail those steps. Now I have drawn out for you these pictures. You can include pictures as part of your notes, or if it's easier for you just to kind of write down the steps. You do have some nice artwork in your textbook, so you can open up to Chapter 10 and find those nice images for neuromuscular junction if you want to have a picture, but you don't want to have to draw it out. Okay, so let's get started. Some background information first. Our central nervous system, which we often abbreviate as CNS, controls skeletal muscle contraction. Now, most of the time, skeletal muscle contraction is under voluntary control, so that means conscious control. That means we're thinking about the types of movements we want to do, and then we produce those movements. I'm thinking about what words I want to say, and so I'm saying those words or my hand gestures and so forth. Also, skeletal muscle can be unconsciously controlled, so our posture is part of the unconscious controlled skeletal muscle movements. We have respirations, which are controlled by skeletal muscle that we're not thinking about. We have lots of other examples. So you train your body to do stuff like walk and write and talk and athletic activities, driving a car, riding a bike. And then we have an unconscious sort of processing that's going to take over. One of the important features of both our neurons, which is our important cell in our nervous system, and our muscle fibers, is that they have excitable membranes. Excitable membranes means that we can have electrical signals. We should more correctly call those electrical signals action potentials. We're going to be talking about the details of action potentials in the nervous system. That's Chapter 11. I may have given the wrong chapter number, but it's coming up, and we'll go through that specific information in the future. So for just some of this information, I'm going to ask you to sort of wait and trust me, and we'll get to that information before the end of the semester. Now, we've already talked a little bit, real briefly, about the resting membrane potential way back in Chapter 3. The resting membrane potential is talking about your cell at rest has a slight negative charge. And the reason it has that negative charge is because of that sodium-potassium exchange pump. Ta-da! You've heard of that before. Sodium-potassium exchange pump, remember, is exchanging sodium and potassium. It is pumping, that means active transport using ATP, pumping 3 sodium out, 2 potassium in to the cell. So you don't have an equal swap of charge. You have 3 positive charged sodium going out, 2 positive charged potassium going in. And then by collecting all of that positive sodium charge on the outside of the cell, anything inside of the cell with a negative charge is super attracted to that sodium on the outside of the cell, and so it's hanging out right inside the membrane. So when we measure it, we have a slight negative charge on the outside and a positive charge on the inside. So we can describe this as being polarized. There's a positive outside and a negative inside. Remember, polarity is talking about opposite ends. So then we have this term called depolarization. Depolarization means remove the polarity, remove that opposite charge. So what we're going to see happening in the case of depolarization, we're going to let that sodium from the outside move in, no longer causing a difference across the membrane. If positive charge moves into the cell, the inside of the cell is becoming positive. So depolarization is talking about the cell becoming more positive. And this can happen until you get equal amount of charge on each side of that membrane. Okay, then we have another term called repolarization. Repolarization means return to that polarity. So this is going to happen by kicking potassium out. If you kick potassium out of the cell goes back to more of a negative charge. So it's kind of reestablishing that negative charge to the cell. Repolarization is talking about making the cell more negative. And then we have a term that's called hyperpolarization. Hyper, means more. So hyperpolarization means more polar. And so in this case, we're going to super duper kick out that potassium so aggressively that our cell will actually be more negative than it was at the start. So hyperpolarization means to make the cell even more negative than resting. Make sure you know these terms. You need to be able to follow the conversation and use them in your own conversations. Okay, now we are going to be looking at our motor neurons, which is a special type of neuron that's going to be talking to or controlling our muscle fiber. It will essentially cause calcium to be released from the SR. Remember, that stands for sarcoplasmic reticulum. And once we get that calcium kicked out of the SR into the sarcoplasm, we will get muscle contraction. So our job here in this video is to look at, well, how does the motor neuron tell the muscle fiber to do this? How does that signal get sent? How does one cell understand what another cell is telling it? How do they talk? So that is our job. That's what we're figuring out here. All of this communication is happening at that structure called the NMJ, which of course stands for Neuromuscular Junction. Now NMJ stands for any neuron muscle connection. So it could be skeletal muscle, which is of course our topic here, or smooth muscle or cardiac muscle. So you'll see it again in the future. Okay, here's my delightful drawing. If you want to try doing the artwork yourself, do a pause and sketch out what you see here, or give yourself a screenshot, or find it in your textbook, or just write out the narrative of what's happening. So it's up to you. The signature of our motor neuron is that a cell body is going to be in the spinal cord, and then it's extending out and synapsing onto a muscle fiber. Now notice those cells don't actually touch. There's space there. It's teensy tiny space, but there's a space, and that space is called the synaptic cleft. Looking too at our skeletal muscle sarcolemma, look at how folded it is. That special folded area is called the motor end plate. That folding is, of course, increasing surface area, so that's significant. All right, inside my synaptic knob, I have vesicles of neurotransmitter. I'm going to see that my action potential is generated from my nervous system, from the central nervous system. It travels down my motor axon, my motor neuron axon, until it reaches that synaptic knob. And I went ahead and drew in on my synaptic knob my vesicles, packages of chemical called neurotransmitter. When the action potential reaches my synaptic knob, that's going to open up voltage-gated calcium channels, and calcium will enter my synaptic knob. Voltage-gated channels are opening or closing based on charge. I like to say open and close because of the gate. Really activate and deactivate are more correct terms. But I'm going to stick with my colorful language there. Okay, so calcium has entered the synaptic knob because of the action potential, and now that's going to cause exocytosis, which of course means a type of vesicular transport of my neurotransmitter, acetylcholine. Big A, big C, little h. Hey, you guys, you're going to see acetylcholine over and over and over and over again this semester. Learn it. It shows up again next semester, so you need to know it. Know the abbreviation, know the full name. Neurotransmitters are chemical messengers that are released at a synapse. Simple as that. This is the primary neurotransmitter we'll talk about this semester. Now that acetylcholine is going to diffuse all the way across that microscopic space called the synaptic cleft, and then it's going to bind to chemically gated sodium channels on the motor end plate. And when it binds, those channels open. Chemically gated means it's a chemical that's acting as the key to unlock the gate or activate the gate. In this case, the key, the chemical, is that neurotransmitter acetylcholine. So once that acetylcholine binds, then our channels open or activate, and it's specific for sodium, so only sodium can move in. Now, where did that sodium come from? Oh my gosh, you guys, you know, sodium-potassium exchange pump has been pumping sodium into the extracellular fluid. All of our body fluids is full of sodium, so it just rushes in. We've created these super big concentration gradients, and so that sodium moves in really fast. Okay, so that big rush of sodium into the muscle cell depolarizes the cell. That's our action potential. So now our action potential has jumped across that synaptic cleft from the neuron to our muscle fiber. So our sarcolemma is excited, and that action potential will travel down into the middle of the cell through those T tubules, and that's continuing all the way across until our muscle fiber is depolarized. So now we've effectively sent our message from the neuron to the muscle fiber. You know, the next part is to look at, okay, what's going to happen with the muscle cell next? And so we're going to do that in the next video. Before we do that, though, let's take a look at how we're going to stop the signal. So ultimately, that motor neuron is only sending that signal for so long. And then we're going to see that the calcium channels close, the neuron stops releasing acetylcholine, the acetylcholine diffuses away from those chemically gated sodium channels on the motor end plate, and then there's a special enzyme in the synaptic cleft called acetylcholine esterase, big A, big C, little h, big E, which will actually remove the acetylcholine from the synaptic cleft, ending the signal, so that we then have control over when that muscle fiber depolarizes and when it doesn't. Okay, that's it for now.